The development of hybrid microcapsules with magnetic nanoparticles represents a cutting-edge advancement in theranostics, a field that combines therapy and diagnostics in a single system. These tiny capsules, often just a few micrometers in size, are designed to encapsulate drugs, imaging agents, or even living cells, making them powerful tools for targeted drug delivery, real-time disease monitoring, and non-invasive treatments. But why is this so important? One of the biggest challenges in medicine today is getting drugs precisely where they are needed while minimizing side effects. Imagine if chemotherapy could attack only cancer cells without harming healthy tissue. Hybrid microcapsules offer a potential solution by allowing drugs to be released only at specific locations under external control, such as a magnetic field or ultrasound stimulation. But how do these microcapsules work? At their core, they consist of biodegradable materials such as polymers or lipids, which form a protective shell around the therapeutic agent. The addition of magnetic nanoparticles, usually made from iron oxide or similar materials, gives them a unique ability. They can be guided to a target site using an external magnetic field. Think of it like a magnet attracting iron filings, but on a microscopic scale, inside the human body. Once the microcapsules reach their destination, they can be activated to release their cargo in response to heat, magnetic hyperthermia, pH changes, ultrasound, or other triggers, ensuring the drug acts precisely where it is needed. An example of their potential application is in cancer treatment. Traditional chemotherapy often affects the entire body, causing severe side effects such as nausea, hair loss, and immune suppression. Now imagine a scenario where a doctor injects hybrid microcapsules into the bloodstream and then uses a magnetic field to direct them straight to a tumor. Once they accumulate at the site, a second stimulus, such as a radiofrequency field, could heat the nanoparticles, triggering drug release and simultaneously destroying cancer cells with localized heat. This two-in-one approach, combining drug delivery with heat-based therapy, could significantly improve treatment effectiveness while reducing damage to healthy tissues. Another exciting application is in real-time disease imaging. Because magnetic nanoparticles are detectable using MRI, magnetic resonance imaging, doctors could track the movement of these microcapsules inside the body, visualizing where the drug is going and ensuring it reaches the intended target. This is especially important for diseases like brain tumors or cardiovascular blockages, where precision in drug delivery is critical. Are these microcapsules limited to treating diseases? Not at all. Scientists are also exploring their use in tissue engineering and regenerative medicine. For instance, they could be loaded with stem cells or growth factors to aid in wound healing or bone regeneration, with their movement controlled externally for precise placement. As research progresses, hybrid microcapsules with magnetic nanoparticles could revolutionize personalized medicine, offering smarter, safer, and more effective therapies. While challenges such as scalability, biocompatibility, and regulatory approval remain, the potential benefits make this a groundbreaking innovation in modern healthcare. What other futuristic medical applications could these hybrid microcapsules unlock in the coming years? Hybrid microcapsules are microscopic spherical structures designed to encapsulate therapeutic agents or diagnostic materials. By incorporating magnetic nanoparticles, MNPs, into these microcapsules, their behavior can be externally controlled using magnetic fields, enabling targeted drug delivery, controlled release, and real-time imaging. These microcapsules typically consist of biocompatible polymer shells that provide mechanical stability while ensuring a safe and efficient interaction with biological systems. The integration of MNPs not only enhances their imaging capabilities via magnetic resonance imaging, MRI, but also enables the use of external magnetic fields for precise positioning and activation. Iron oxide nanoparticles are commonly used in hybrid microcapsules, allowing for simultaneous drug delivery and MRI-based tracking in cancer therapies. These microcapsules function like magnetically guided submarines, traveling to a target destination a tumor while carrying essential cargo drugs and transmitting real-time location data for monitoring. Magnetic nanoparticles exhibit super paramagnetic behavior, meaning they respond strongly to an external magnetic field but lose magnetization once the field is removed. This property makes them ideal candidates for theranostics, a combination of therapy and diagnostics, since they can be used for both drug delivery and non-invasive imaging. Functionalization with biocompatible coatings, such as pegylation or silica shells, further enhances their stability and targeting specificity. In addition to MRI contrast enhancement, MNPs can also be utilized in hyperthermia therapy, where localized heating induced by an alternating magnetic field destroys cancerous cells. Super paramagnetic iron oxide nanoparticles, SPIONs, are widely used in clinical research for detecting tumors and delivering chemotherapeutic drugs with magnetic guidance. MPs act like smart thermoregulators in a heating system, activated only when needed. They generate localized heat to treat specific regions without affecting the surrounding environment. Microfluidics offers precise control over the synthesis of hybrid microcapsules, allowing for the uniform encapsulation of MNPs and therapeutic agents. Using droplet-based microfluidic devices, researchers can generate monodisperse microcapsules with customizable sizes and compositions. These systems utilize flow focusing or TJ junction techniques to control the encapsulation process, ensuring high reproducibility and efficiency. The ability to fine-tune microcapsule properties, such as permeability, degradation rate, and magnetic responsiveness, makes microfluidic fabrication a promising approach for theranostic applications. Droplet-based microfluidic synthesis enables the production of core shell microcapsules loaded with deoxyrubicin, a chemotherapy drug, and iron oxide nanoparticles for enhanced tumor targeting. Microfluidics operates like a precision bakery, ensuring each capsule is perfectly formed, consistent in size, and filled with the exact amount of active ingredients. One of the major advantages of hybrid microcapsules with MNPs is their ability to be directed towards specific locations in the body using external magnetic fields. This targeted approach minimizes systemic side effects by reducing off-target drug accumulation. Once the microcapsules reach the desired site, PN, a tumor or inflamed tissue, localized drug release can be triggered using heat, pH changes, or enzymatic reactions. This enhances therapeutic efficacy while reducing the required drug dosage. Magnetically guided drug carriers have been used in preclinical trials for targeted cancer therapy, significantly reducing side effects compared to conventional chemotherapy. The process is akin to using a GPS-guided delivery system, ensuring that the package reaches the correct address, target tissue, without unnecessary detours. Magnetic hyperthermia is a technique that exploits the heat-generating properties of MNPs when exposed to an alternating magnetic field. When hybrid microcapsules containing MNPs accumulate in tumors, they can be heated selectively, causing localized cell death while sparing healthy tissues. This technique is especially promising for treating drug-resistant tumors and deep-seated cancers. Additionally, the mild heating can enhance the permeability of tumor blood vessels, improving drug absorption. Clinical studies have demonstrated that magnetic hyperthermia in combination with chemotherapy significantly enhances treatment efficacy in glioblastoma patients. This method is like a precisely controlled microwave oven, heating only the intended target while keeping the surrounding areas unaffected. Magnetic nanoparticles enhance MRI contrast by shortening relaxation times in tissues, making them ideal for real-time imaging of drug delivery. When encapsulated within microcapsules, they allow non-invasive tracking of therapeutic agents within the body. This helps clinicians monitor drug distribution, assess treatment response, and adjust dosages accordingly. Advanced imaging techniques such as dual model MRI and fluorescence imaging further improve accuracy. Spine-loaded microcapsules are used to track the movement of stem cells post-injection, ensuring they reach the target site in regenerative medicine. MRI tracking with MNPs is like using night vision goggles in a dark room, enhancing visibility to locate otherwise hidden structures. Hybrid microcapsules can be engineered to release their contents in response to specific physiological triggers. Temperature-sensitive polymers allow heat-induced drug release, while pH-reinvestitive materials enable controlled release in acidic tumor environments. Magnetic field-assisted disruption of capsule shells provides another method for precisely timed drug administration. By integrating multiple stimuli resupply mechanisms, researchers can create smart drug carriers that optimize therapeutic effects while minimizing toxicity. pH-sensitive microcapsules have been developed to release insulin in response to glucose levels, improving diabetes management. These systems work like self-assessing locks, only opening when the correct environmental conditions are met. Beyond drug delivery, hybrid microcapsules incorporating MNPs hold promise for regenerative medicine. They can serve as scaffolds for cell growth, guiding tissue repair through controlled magnetic manipulation. In bone regeneration, magnetically responsive capsules loaded with growth factors can be positioned at injury sites to stimulate healing. Similarly, in neural tissue engineering, magnetic cues can be used to direct stem cell differentiation. Magnetically guided microcapsules, loaded with bone morphogenetic proteins, BMPs, have been used to accelerate bone repair in orthopedic applications. These microcapsules function like construction workers following a blueprint, delivering necessary materials to build and repair damaged structures. Despite their potential, several challenges must be addressed before hybrid microcapsules see widespread clinical use. The biocompatibility and biodegradability of materials must be optimized to prevent long-term accumulation. Precise control over drug loading and release kinetics remains a key area of research. Additionally, large-scale production and regulatory approval require extensive validation. Future advancements in nanomaterials, artificial intelligence-driven drug delivery, and personalized medicine approaches are expected to enhance the clinical translation of these technologies. Current studies focus on using biodegradable polymers such as PLGA, polylactic coglycolic acid, to ensure safe degradation after drug release. Developing these microcapsules is like designing next-generation spacecraft, balancing precision, safety, and efficiency for long-term success. Hybrid microcapsules incorporating magnetic nanoparticles represent a breakthrough in theranostics, combining targeted therapy with real-time imaging capabilities. Their ability to deliver drugs with precision, enable hyperthermia treatment, and enhance MRI contrast makes them powerful tools in modern medicine. While challenges remain, ongoing research in biomaterials and microfluidic synthesis techniques will further refine these microcapsules, leading to safer and more effective treatments in oncology, regenerative medicine, and beyond.